So our next speaker is actually going to address this subject. A good friend of mine, Jeff Kaufman, who has undertaken, I think very successfully, the mission of creating science to address a very unusual but a deadly problem at times, and that is adenoid cystic tumors. And Jeff's going to tell us how he did it.
First of all, I'd like to thank Bruce so much for inviting me to speak before this very distinguished audience. In this talk, I'd like to give an overview of our 5-year journey. We've been, actually next month will be 5 years that the Adenoid Cystic Carcinoma Research Foundation has been in existence. And I want to talk about how we've invigorated what had been a backwater of rare disease research by developing biospecimen resources and new animal models that didn't exist before, by identifying molecular targets that were specific to ACC, by carrying out preclinical screening of targeted agents, and providing what is the endpoint of all that we're doing, which is hopefully a sound scientific rationale for moving forward with clinical trials that have a high likelihood of success in ACC. So hopefully I'll give you a sense of the challenges and the opportunities in rare disease research and make the optimistic case that hopefully patient-coordinated and assisted research can accelerate progress. So first, oops, no, I'm sorry, can we go back one slide? Yeah. So quickly on statistics, I got to get them out of the way. Rare diseases, defined by the Rare Disease Act of 2002 as the diseases afflicting fewer than 200,000 Americans. The paradox is that rare diseases are actually very common. There are about 30 million Americans, 10% of the population, that have a rare disease, one of 7,000 rare diseases. Individually, no rare diseases that common, clearly, but there are just so many of them. Narrowing down on rare cancers, if you leave out about the top 10 or so cancers that don't meet the definition of a rare disease, you're still left with an enormous healthcare burden. 25% of all cancer deaths are in rare cancers. 27% of all cancer diagnoses are in rare cancers. We know cancer is the leading cause of death heart disease is the leading cause of death in the United States. Cancer is close behind. The thing that really struck me is that if you carve out rare cancers from those top 10 or so common cancers, rare cancers actually would be the third highest cause of death in the United States, about 5.8% of mortality. So I just, we say rare, but it really is an enormous healthcare burden that this group of cancers have on our country. But I am optimistic about a lot of, about the prospects for rare disease research and rare cancer research because there's been a shifting drivers of rare disease research. The traditional view has been that rare diseases don't have enough available specimens, there aren't preclinical models available, it's too hard to organize the few patients and interested researchers, it's too expensive to carry out the research, not profitable enough for private industry to focus on, in that innovation really spills over from common diseases to rare diseases. You just sit around hoping that there's a drug that works on a common disease that's going to work for your rare disease. The reality has shifted dramatically over the last 10 years, I think. To the point where now the internet permits organization of interested parties not only for raising funds, getting patients together, but also for organizing researchers. Levi Garraway yesterday had a chart that was great that showed the cost of sequencing plummeting, in fact, more rapidly than the cost of semiconductor power, Moore's law. So we are seeing that Moore's law does apply to a lot of basic research, for cancer. Everyone talks about personalized medicine, and really what's underlying that is is that even those 10 most common cancers really are just an agglomeration of lots of subsets of rare cancers. So all cancers are rare cancers at this point. The field is moving towards us, and actually it's in a very positive way for rare cancers, because we're looking for those hills that Levi was speaking about. And clearly pharmaceutical companies get that structural shift and are targeting smaller and smaller patient subsets. They're seeking orphan drug designation more and more. And I think it's better understood now that innovation often can spill over to common diseases from rare diseases. I mean, the classic case is with the rare familial cholesterol diseases, where the advances in the knowledge about the basic biology of cholesterol then opened up blockbuster opportunities for cholesterol drugs. So quickly looking at some statistics on orphan drug designation, there are over 360 drugs with orphan drug designation that have been approved by the FDA for about 200 rare indications since 1983. Interestingly, over the last 2 years, about 1/3 of all new molecular entity approvals had orphan drug designations. And if you look at biologics, it's 2/3 of those. So clearly, there's some very powerful forces that are shifting the way research is being done, and it's seeping not only through the basic research but through to the pharmaceutical companies. So now I'd like to, with that backdrop, I would like to talk about ACCRF. Our experience. I want to have a very, very quick review of the nature of the disease that we're dealing with, adenoid cystic carcinoma. It is a malignancy of the secretory glands, most frequently found in the salivary glands, but also mammary, lacrimal, Bartholin, sweat glands. About 1,200 new cases each year, prevalence of about 10,000 patients in the United States. It's not inherited, it's not associated with ethnicity, tobacco, alcohol consumption, viral infections. Slightly more women than men get ACC. And it does strike patients about 10 years, the median patient is about 10 years younger than the median patient for all cancers. You get a lot of young mothers in their 30s, 40s, 50s getting ACC, as was the case with our family. It has a very high propensity for perineural invasion and late metastasis to the lungs. So you get 5-year survival rates that are actually quite high, 70–80%. But the recurrences are frequent, and so when you get out to 10–15 years, you get below 40% survival. The standard of care is just surgery and radiation. There have been 25 phase 2 clinical trials in ACC, all with very, very small patient numbers, and it's very chemo-resistant. There's no effective or approved drugs for ACC. Quick background on our foundation. We've only been around nearly 5 years. After my wife's diagnosis, I just wanted to write a check to someone and say, here, go do research. It just wasn't available. There's no, there wasn't any entity that you could give it to that would do the research. My wife and I decided all we could do was try and drive it forward ourselves. The nature of ACC is such that you do have that window in which it's slow growing enough that you can make progress, but it is serious enough eventually that you want to make progress. So our strategy was threefold. We wanted to build a community of interested researchers that, with the help of our scientific advisory board, would create an innovative research agenda, and that we would mobilize patients in support of that agenda by providing specimens, by giving funds for research, and by enrolling in trials. So looking at our research agenda, we had 5 guiding principles that really drove everything we did. The first is, we're here as venture philanthropy. Our goal is to bridge the valley of death, to get enough of a good scientific rationale in the basic and preclinical translational research to take it to the point where private industry or CTEP will then move on with clinical trials. So that's our job, is to give good, clear scientific rationales for why a drug should work particularly well in ACC patients. I don't know, for Monty Python fans out there, that's actually the bridge of death that we often feel like we're crossing across the valley of death. But I mentioned before my background is as a portfolio manager. My view of research projects is if there was any individual project we knew was going to give us the answer, we would put all our resources into it. That's not the way either finance works or medical research works. What we're looking to do is create a portfolio of projects, each of which is effectively a stone in a mosaic. We're trying to get signals from all these different research projects, integrate those individual stones, and build together a mosaic that gives a fuller picture of the entire disease. We're also a small foundation. We don't have the luxury of passively sitting back and reviewing investigator-initiated proposals. Our scientific advisory board has been wonderful in proactively setting the agenda, saying, we know these are the projects that we have to undertake and get done, and we're going to go out and find the investigators who have the expertise, have the interest, and the capacity to carry out those projects. And so that's been a huge amount of my time is proactively going out and selecting, having projects selected by our scientific advisory board, but then implementing them, making sure they happen. No single institution can do all of those pieces, certainly not for rare diseases. So it's very important that we get a multi-institutional group that plays very, very well together in the sandbox. And that's difficult. It's easier for rare diseases because everyone knows they can't do it themselves. And lastly, we do focus on accelerating practices where we can own the data or own the resources, xenograph models or whatever. We do, but we require of our investigators to rapidly share their data, share their resources, and it's been fabulous. We've got a wonderful group of investigators. So now quickly, here's a schematic of our research agenda that it hasn't changed a great deal over the last 5 years, but basically it's focused on three different categories of projects. The first of which is specimens and models. For most of the diseases that you all study, it's presumed that whole set of resources are available. It's not the case with rare diseases. And in adenoid cystic carcinoma, we spent most of the first 2 years just trying to get biobanking projects up and running, create the xenographs. So that took a huge amount of time, and basically getting some raw material with which researchers could carry out their studies. The second category, the second stratum, is target and pathway identification, answering the question, what targets matter in ACC, specifically in ACC. And we've undertaken projects at a wide range of institutions. DNA sequencing, Andy Futrell at the Sanger Institute is just now finishing up an exome sequencing project, and also paired end sequencing to identify rearrangements in ACC. We've also funded various ACGH studies. Steve Balin at Johns Hopkins has finished a methylation study. All just on down the line. And our concept has been that what we want to do is have a consistent set of specimens and models fed through all of these different high-throughput technological platforms. So we're generating data sets from the same samples with across high-quality platforms that will eventually get to this bioinformatics area where we're going to try more and more to integrate the data. And that's our eventual goal. I won't say that we're there by any means, but that's the framework in which we're working. And then lastly, on this third stratum of projects is translational research. It's, for this audience, I apologize, it's comically simplistic. And so, we've tried to get the targets that have been thrown up by that second tier. That second tier of projects, and then bring in drugs and screen them in our xenograph models and to get some preclinical data on the efficacy of both approved and novel drugs. One additional point I'd make on this slide is that we've started all these projects. We've brought together all these investigators for ACC, but we've developed great relationships with the NIH. The National Institute of Dental and Craniofacial Research, NIDCR, has taken a strong interest in salivary gland normal development and cancers. Larry Tabak was just promoted from the head of NIDCR to deputy director, I believe, and he's a fervent leader in what he calls, I believe, the salivation army, which is particularly heartwarming for us. But they've actually come and partnered with us, where we've gathered investigators for a biobanking project. NIDCR has backfilled that for not only ACC but all salivary gland tumors. So they've given $5 million for that project. We had started a project in which we were developing xenograft models, low passage, fresh human tumor xenograft models. Again, NIDCR came along and funded 2 challenge grants to develop xenograft models for all salivary gland cancers. So they're coming and backfilling the effort, the groups that we've seeded and broadened them. Likewise, they're doing that with a lot of the genomic and proteomic studies that we started at the Sanger Institute and other institutes. Well, I think I've mentioned some of these institutions, but this is our community of researchers that has developed. We've got folks at the University of Virginia, MD Anderson, Hopkins, the Farber, Mass General, cell signaling, UCSF, even Sweden, so Gothenburg University. So this is how I spend my days, is trying to build this community. The history has been very focused on that basic research, and it's taken me a long time to try to understand the complexity of what you all do every day. But as we're moving closer and closer to the clinic, I'm finding that the complexity is even greater, and I have an even steeper learning curve for understanding clinical trials. And I look at the 25 clinical trials that have been done in ACC, and honestly, there's not a single, there's one of the 25 where I could say that I understand the scientific rationale for why that drug should work in ACC. The rest, honestly, it's crop dusting. There are too few ACC patients out there that we can't waste that valuable resource without having intelligently targeted drugs. I mean, it's particularly in rare cancer communities. We need personalized medicine in order to take advantage of the few opportunities we have to find better medicines. So this is a framework that I've put together, try and educate myself and other patients as we go out and try and increase trial accrual, and it's putting together rationales for why a drug should work just in ACC or our particular disease, and I've grouped them in 3 broad areas: mechanism of action, which is really Bruce, who's on our SAB, has always driven that point home. Let's focus on the basic biology of the disease. Let's understand the mechanisms of action, and that's going to be the highest quality projects and the highest return projects that we can invest in. And in there, but even within mechanisms of action, there are very, very different levels of evidence there. A lot of there was great hope that imatinib would work in ACC because 90% of ACCs overexpress c-KIT. But there are thousands of genes that are overexpressed in ACC, and it's great that there's overexpression of c-KIT, but later studies that confirmed that there's no phosphorylation of c-KIT in ACCs, and then when we tested the drugs in our mouse models, it confirmed what we already knew from the clinic, which is that this isn't a drug that's gonna impact ACC patients. So what we're looking to do is build additional layers of certainty that a drug is going to work. So we're looking at phosphorylation, identifying DNA alterations, and eventually getting to the functional studies that will lay out the pathways and networks that matter specifically for ACC. We also have set up a platform for preclinical screening, and the reality is that, so the top two categories are really the ones in which we have the greatest faith. The fact is there are partial responses in phase 1 trials, and there are some trials that have moved forward based on that, and certainly we want to follow up on those, and we have. So just to summarize it, the simplistic clinical trial math that's in my brain is simply, we're here to provide strong scientific rationales for clinical trials. We're going to provide supporting preclinical data, and out of that, you're going to hopefully get excitement from industry to proceed with the trials. Just as importantly, you're going to get excitement among the patient community to enroll in those trials, and that's how we're going to be successful. So now I just spent a few minutes on the levels of evidence, particularly mechanisms of action in ACC. And as I said, we've generated a lot of these data sets, and we're just now starting to try to integrate them. One of the simplest things you could do is a Venn diagram of look at what are the targets that are overlapping in the various data sets, and we have identified FGFR1 is a target of interest, IGF-1R as well, and our hope is that we're going to undertake much more sophisticated analyses going forward. What was nice is that these were unbiased studies. You looked at all gene expression. We looked at the whole kinome, actually the tyrosines, I'm sorry, for the phosphorylation. And that's how we came up with those targets. Totally in parallel, we had another funded researcher in Sweden who identified a recurrent translocation in ACCs. And it basically is a T69 translocation, which involves the MIB gene, which is a known oncogene, and NFIB. And what happens is the negative regulatory domain of MIB is lost in the fusion, and so these microRNAs that dock and stop, downregulate MIB are no longer there. So MIB is one of the most overexpressed genes in ACC, So we've funded studies to identify the prevalence of this fusion across much larger cohorts. And it looks like it's probably going to be a solid majority of ACCs are going to have this fusion gene. But interestingly, even the fusion negative cases of ACC also overexpress MIB. So there may be an alternative mechanism by which MIB is overexpressed. Functional studies are underway, and also we've got some efforts to create transgenic models. Kwok Wong at the Farber, Gigi Lozano at MD Anderson, and Inder Verma at the Salk Institute are all involved with different approaches to try and create transgenic models of ACC. So we believe, it's by no means certain, but we think we found the likely driver of ACC, and so the comprehensive long-term strategy is to try and go after the driver, which is MIB, but it's a transcription factor, very difficult to drug. We are exploring a staple peptide approach, but in the interim, we have to follow an opportunistic strategy and look at the downstream targets of MIB. There are 16 known downstream targets, 14 of which are overexpressed in most ACCs. So that's kind of where we're focused on. And sure enough, one of the targets is FGF2, which is ligand for FGFR1, which we had identified from our unbiased studies previously. So that was encouraging. But we need to identify more downstream targets. We're going to do ChIP-seq studies and so forth to do that, and sort of methodically work our way through the rational targets. Quick point on preclinical models. When we started the foundation, I gathered all the cell lines that I could find that were published in the literature. We disseminated them to our community of interested researchers. I got back from 3 separate researchers comments that they'd undertaken STR profiling. It turned out all of the cell lines were misidentified. We had 3 HeLa cells, a bladder cancer cell line, there was a murine tumor in there. So I was devastated at the time, but the fact is we cleaned up the field. The fact that there are all sorts of conclusions that were being made about ACC that were invalid, and the good news is that at least those aren't proceeding. We did also in parallel focus on xenograft models, creating these models, and what we've done is we've hired a CRO to centralize them all, to expand them, establish them, and we've completed 5 baseline screens of 40 anti-cancer agents across all those models to try and get a sense of, well, how sensitive or insensitive are each of these models, and then we can evaluate novel agents against those baselines. So this is a summary. It's an average tumor growth inhibition across all 5 of the baseline models that we carried out in our xenograft models. This is only the approved agents. We couldn't put in the novel agents, but you can see that actually the only docetaxel shows up as really significantly active on average in our models. The rest are pretty much in line with what we've seen in the clinic, which is that they're really, these models are resistant to most of these therapies, just like in the clinic. I know I'm running late here, so let me hurry. So trying to tie it all together, what are we trying to do? What we want to move forward are the highest, those clinical trials that have the highest likelihood of success, that have the highest levels of evidence. So what I've listed here are actually there are 8 clinical trials that are open for ACC patients at the moment, and what we've done is sort of graded them across mechanisms of action, preclinical screening, and clinical anecdotes. And what you see is that the 25 studies that were done previously, but even the current studies that are open, there really is not a clear scientific rationale for why any of these drugs should be working in ACC. We've been in discussions based on our basic and translational research, getting in drugs from various companies and screening them in our xenograft models, and we're moving forward with a trial in Dovitinib, which is an FGFR, well, it's a multi-kinase inhibitor, but it includes an FGFR1 as one of its targets. And by no means do I feel any certainty that this is definitely going to work, but I just think that the likelihood of success is right. It is higher. This is the approach that we want to take. We think it's right for the patients. It's right for private industry. This is the way that we're going to move forward in the best manner. We do want to continually improve our continuous data improvement. We're generating these data sets, and we're trying to integrate them all. But just one thing that was interesting from our xenograft data is here I have the tumor growth inhibition across these 5 ACC xenograft models, and we've got the top performing drugs on average. And that's interesting, and definitely I think docetaxel might be worth looking at in the clinic. But also what's perhaps more interesting is if you look at the average TGI of sorafenib was only 45%, not very impressive at all. But what would be more interesting is to look at those cases. There were actually 3 cases in which there was pretty good activity in sorafenib, and 2 cases where there was actually no activity. And so the type of studies we want to undertake is to try and correlate the drug efficacy with the genomic profile, and that's sort of the wave of the future of what we're hoping to do. The other thing that we're going to do with all these data sets that we're generating, there's already a sort of pilot data set sitting on the Sanger's Cosmic portal, but over the next couple of 3 months, the exome sequencing data across 25 ACCs is going to be put out there, and so that will all be publicly available. The University of Virginia has a also very extensive data set for the xenografts in some primary tumors. That's also out there and will be is widely available. So our hope is that put the data out there and let 1,000 flowers bloom. Let people work with the data. So just in conclusion, we've spent 5 years almost and about $3 million, and what did we get for it? I think we've actually gotten quite a bit for what I would view as relatively modest investment. in a rare disease research. We've gotten over 200 frozen specimens in our biobanks. We invalidated all the pre-existing cell lines. I guess that's an advance. We've characterized and created and characterized about 15 human tumor xenografts and completed 5 baseline screens. We've gotten NIDCR to co-fund with us $12 million in projects. And we've identified a likely driver translocation. We're in the process of creating transgenic mouse models, generating a ream of genomic and proteomic data. And we've gotten to the point where I think we have the first clinical trial in ACC with a clear scientific rationale supported by preclinical data. Lessons for rare disease research. I do think it's important to try and organize a community of interested investigators. I think as patient advocacy organizations, we have a special place in which we're able to use our moral persuasion and our full-time focus on it. Even looking at our community, maybe there's 3 to 5 investigators who spend 25% of their time on ACC. We don't have anybody other than us that's full-time on ACC. So I think for rare diseases, you need that central gathering point. The second point I'd make is you got to be friendlier to the pathologists. They are just so crucial to everything that's going on, to get the specimens, but also to analyze them, to later for the diagnostics and so forth. I think that I look at the way that the relations between pathology and other disciplines is at a lot of institutions, and we've been just very, very fortunate that we've got a very collaborative group, and I think that's been essential to everything. As I said before, the primacy of focusing on the mechanisms of action, that's crucial. And the other thing that I found is in talking to private industry, they're not here to cure ACC. They're here to talk about targets, and that's the way you've got to approach rare disease research. Is not to say, oh me, oh my, please, help me. You've got to be hard-nosed about it and say, look, these are the targets that matter in our disease, and this is the rationale why you should move forward. So final slide, it's just, you all put up the slides of the acknowledgements, and we don't have a lab. I don't personally have a lab, but I do have the heroes that we're very, very thankful for everything that they've done. And our scientific advisory board has been absolutely so committed, so helpful, and Bruce has been fabulous in that, in giving us guidance, so we appreciate it. And we've also gotten an extended community of probably about 20 to 30 researchers, but there really is there are 6 cornerstones of our research effort, and you can see their names up there, and we're so appreciative of all their help. So thank you very much.
Maybe a question or two.
Sorry, I ran over.
It's very interesting, very important.
I think it's a very important exercise, what you're doing, bringing these people together in this rare disease. Now, you picked ACC today, and that's a disease where when it's metastatic, it may grow very slowly or not grow for 10 years or 15 years. And it would be very important with your many centers or the people involved to try to understand why these metastases don't grow. You are doing genetic studies in these tumors, but there must be a reason why they don't grow, and I think it would be worthwhile for other diseases to understand why this disease does grow so slowly.
Yeah, no, I agree, and it could well be that there's a lot of the studies on senescence and late metastasis that we need to focus more on and understand better. And it'd be interesting whether it's ACC specific or applicable perhaps to all cancers in the same way. So that could be a way in which we learn something more broadly about other more common cancers.
Jeff, congratulations on the vision and leadership that you've shown, because I think what you've achieved in 5 years is absolutely outstanding. You didn't talk about education as an important component of the foundation, and I'm just going to pose the question as someone who remembers the first time as an attending physician he came across the patient with adenocystic and scratched his head and went, where the hell's that in the literature? and the problem for clinicians and for oncologists, indeed surgeons, is that they very rarely see these patients. And my view would be that both the physician and the patient are lost when this diagnosis happens initially. So I just wonder whether the foundation has thought about the whole idea of instructing the patient and the physician as to what the best approach may be. You talked about a joined-up clinical trial strategy, because I remember when I reviewed the literature 15 years ago, the largest collection I could find, I think, was a memorial with 10 patients. So maybe could I hear your thoughts on that?
Yeah, no, absolutely, and it's what we're actually on the cusp of doing is putting out some more educational materials for well-informed patients, but also for physicians who haven't run across ACC very often. And actually, in the next week or two, we're going to have a document out that will review the history of all past clinical trials, the currently open trials, and potential molecular targets. And the whole point is education. So, we realize it's a
I don't know if my microphone is on, but it's a remarkable document. I've read it, and I just am in awe of what Jeff has put together as a layperson. If he knew that much about bonds as he knows about ACC, he would have been a remarkable financial advisor, and I'm sure he was. I think we should think, actually, Jeff, if you're amenable to this, about putting this online as part of your presentation, and we could do that because I think it's the best, most comprehensive discussion of the disease and its potential for therapeutic intervention that I've seen.
In fact, Bruce, that was going to be my point. Why is it you take my point? Jeff, it's true, we do. Jeff, truly, I was just going to not only suggest about putting this virally online, but also one of your greatest advantages, I should think, of recent years has been the friendship that you must have with Amy Doxer-Marcus of the Wall Street Journal, who the Pulitzer Prize-winning journalist for her series of articles, front page 1 in the Wall Street Journal on cancer in general, until her mother was diagnosed with a rare cancer, that then her editor said, we want you to look into and do something about. Subsequently, as you know, she became a fellow and went to the NIH and so forth and so on. But there is now, through the media, is my point, an opportunity not only to express the pathos and significance of the individual patient, however few they may be, but to say this is a marvelous and incredible opportunity of experience to learn from these unusual diseases, but in great depth for a host of other reasons as well.
Yeah, I would just say, yeah, Amy is actually a nearby neighbor of ours and has become a very close friend. So you're absolutely right. But I also don't want to say that we're the only sort of research-oriented patient advocacy group out there. There really is a grassroots bubbling up of similar organizations like the Chordoma Foundation, the Caring for Carcinoid, and others, where there are patients who really are looking to grasp the reins, not in a confrontational way, but in a supportive way as we can possibly be, of the wonderful work that you all are capable of doing. And that's, we want to make it easy for you to look across, a broader range of diseases, including the rare diseases. And absolutely, we will be raising the profile over time.
I would just invite everybody who's interested in this subject to talk to Jeff over lunch. I'm sorry we don't have enough time to continue the conversation, but thank you so much, Jeff, for the presentation.